Okay, so we left off talking about genetics, and I wanted to go over some terms really quickly that I just kind of threw around a little bit last time, and I wanted to define them a little bit more explicitly and talk about how it is we use these terms. So the first two terms are gene and allele. A gene is just a non-random sequence of DNA that codes for some character. So we can talk about a gene for eye color, and a gene for hair color, and a gene for creating a specific enzyme or a specific protein. So a gene is just a region of the DNA that codes for something, some character. An allele is an alternate form of that gene. So when we say that there is a gene for eye color, it doesn't tell us what eye color actually is, right? Eye color is actually determined by the alleles that we have at that gene locus. So for eye color for us, there is a gene, eye color, and then there is the allele that is brown, and then there's the allele that is green. And those are really the only two eye colors. There are blue eyes as well, right? But we'll talk about that a little bit later. That's kind of a different sort of scenario. In terms of Mendel's experiments, there was a gene for flower color, right? And the alleles were either purple or white. We also talked a little bit about genotype and phenotype. So a genotype is just the actual alleles that you have. So purple plants had the genotype of capital P, or capital P, lowercase p. And white plants had the genotype of lowercase p, lowercase p. So when I refer to a genotype, really what I'm looking for is not the word purple or white. What I'm looking for is the actual alleles, two capital P's, a capital P and a lowercase p, or two lowercase p's. And then a phenotype is just the physical manifestation of those alleles. So for Mendel, it was purple or white for flowers, right? For us, it's either green or brown eyes or hazel eyes or blue eyes. That's the phenotype. So what Mendel did was he generated some ratios, both genotypic and phenotypic ratios. And the first phenotypic ratio that he generated was between the two F1 generation individuals. Remember, he took two true breeding lineages and he crossed them. That was the parental lineage. And he crossed them and he got an F1 lineage. He took a purple flower and crossed it with a white flower. That purple flower could only produce purple offspring. White flower could only produce white offspring. He crossed them, and then all of the offspring were what? They were all purple, right? That was the F1 generation. The first filial generation was all purple. So he took that first filial generation and he crossed that. And what we decided was that first filial generation had the following genotypes. So we took an individual that looked like that, genotype capital P, lowercase p, and crossed it with another capital P, lowercase p. Those two individuals have what kind of phenotype? So that's the genotype. That's a good way to describe the genotype. But what's the phenotype for this heterozygous individual? Purple, right? So two purple individuals that are exactly right, heterozygous. And the way that we diagrammed that cross was with a Punnett square, right? And remember that a Punnett square is just a really convenient way for us to model meiosis and fertilization. And what we're saying is, Dad could produce a gamete that has a capital P in it, or Dad could produce a gamete that has a lowercase p in it, right? That is, Dad could do that or that. Mom can do the same thing. She could produce an egg with a capital P, or she could produce an egg with a lowercase p. That goes there, and that goes down there. So if this pollen grain fertilizes that egg cell, then this individual's genotype is what? Capital P, capital P. And we gave that capital P a special name last time. Homozygous dominant, exactly right. Okay, so then if this pollen grain fertilizes that egg cell, the genotype is capital P, lowercase p, and then as Ashton said, that's heterozygous, right? Same thing right here. capital P pollen grain fertilizes a lowercase p egg cell, we get another heterozygous individual. And again, same genetic convention, right, with the capital going first and the lowercase going second, or dominant first, recessive second. And then this final individual has what gene? Homozygous recessive, right? Lowercase p, lowercase p. That's that individual's genotype. And the phenotype is white, exactly right. So what is the ratio of purple to white? 3 to 1. Exactly right, because each of these has a dominant allele, each of them will be purple. This one lacks a dominant allele, so that recessive character is expressed, right? So there are one individual in that cross. Okay, so what is the genotypic ratio?
1 to 2 to 1.
1 to 2 to 1. That is homozygous dominant to heterozygous to homozygous recessive. So those were Mendel's first two genetic ratios. So you guys, if ever you cross two individuals that are heterozygous, these are always going to be the genetic ratios, the phenotypic and the genotypic ratios. Does that change if we change the genetic structure or the genotype of one of the parents? The answer is absolutely, right? So what happens if we cross an individual that's, let's say, heterozygous with an individual that is homozygous recessive? What proportion of those offspring are going to be purple, and what proportion are going to be white, do you think? You think it's going to be 3 to 1 still? Okay. You think it's going to be 3 to 1?
Actually, it's 4. So you said the phenotype?
Yep. How many are going to be purple and how many are going to be white?
They're all going
You think they're all going to be purple. So everybody's going to have a purple allele.
oh, sorry. I thought they were all. 2 to 2.
2 to 2. And we can reduce that to 1 to 1, a 1 to 1 ratio, right? Let's see. So we have a couple of hypotheses. We have a 3 to 1. We have a 1 to 1. So if this is mom up here and she is homozygous recessive, she can only produce eggs that carry the recessive allele, right? Dad happens to be heterozygous. So what color is mom? White. Dad is purple. Okay. So dad can give up a dominant allele, and he can also give up a lowercase allele. And I know this is confusing because the capital and the lowercase p look very similar. I should have used a different letter like A or something like that. But anyways, this dominant pollen grain fertilizes that recessive egg cell. What's the genotype? Heterozygous, right? Capital P, lowercase p. How about right here? Same thing. How about right here? Lowercase p, lowercase p, right? So homozygous recessive. And then this one? Same thing. So we have a phenotypic ratio of purple to white of 2 to 2, or we can reduce that to 1 to 1, right? What's the genotypic ratio? It's the same thing. 2 heterozygous individuals to 2 homozygous recessive individuals. It's a 1 to 1 ratio. Okay? This is actually called a test cross. We're going to talk about a test cross in just a minute. Okay, so let's get back to this. This was Mendel's second ratio. I'm sorry that the white just kind of bleeds out. Basically what it's saying is, in this cross, what we get is a 1 to 2 to 1 genetic ratio. One individual that is homozygous dominant, or using Mendel's terms, true breeding purple, to 2 heterozygous individuals that are not true breeding, to 1 individual that is homozygous recessive, or true breeding white. Okay? Okay.
How do you know if people are?
How do you know if people are? We're going to talk about some genetic characters for people, and that's super interesting. So let's stay that conversation for just a few minutes. From this one experiment, Mendel came up with a number of hypotheses. And this is, again, considering just a single character in isolation. The first hypothesis suggests that parents don't actually transfer their characters, rather they transfer information about their characters. Now, that makes some intuitive sense to you and me, because we understand what DNA actually is, right? It's just information. But back in Mendel's time, people didn't think that. People thought that parents in their gametes actually gave portions of their characters. That is, mom gave up some of her green eye, and dad gave up some of his brown eye to have a hazel-eyed offspring. And what Mendel says is, no, that's not true. What's actually happening is there's just information about those characters that are being transferred between generations. And that information is called a gene. And that gene, again, is a non-random sequence of DNA with a specific locus, a specific location. Now, again, please remember that Mendel didn't have the same molecular techniques that we have. He couldn't isolate DNA. He was looking at whole organisms. And from looking at these whole organisms without a microscope, he comes up with some of these really great hypotheses. The second hypothesis is that every individual, and in the context of parents, both parents have 2 copies of each gene. And that's true, right? We know that to be true. That's what the foundation of this idea of homologous chromosomes actually is. Each of us carries 2 copies of each gene. And there are 2 classes of genotype. You can either be homozygous, that is, both of your alleles are the same, or you can be heterozygous, and you have one of each type of allele. This also suggests something else, you guys, that for every genetic character, there can only be 2 alleles. That is an inherent assumption with this hypothesis. What we're going to find out later is that that's not necessarily true. Hypothesis number 3 is that this idea of alleles, it's alternate forms of genes that actually code for the phenotypic characters that we see. And that alternate form of a gene is called an allele. That allele determines both what our genotype is, and the relationship between those alleles determine what our phenotype is. Hypothesis number 4 states that an individual has 2 alleles, and that those 2 alleles act independently. Additionally, those 2 alleles affect just a single character. So there are 2 ideas here. Alleles are independent, that is, one gene really affects one phenotype. You betcha. One gene essentially affects one genotype. One gene affects one phenotype, excuse me. And conversely, one phenotype is only controlled by a single gene locus. What that means is eye color for us has to be controlled, at least according to Mendel, has to be controlled by a single gene. It can't be controlled by multiple genes. Flower color in peas can only be controlled by one gene. And that gene for flower color can only control one phenotype. Flower color can't also affect seed color, seed shape, or anything like that. They're all independent, right? So for every character we can contrive within our own bodies, according to Mendel, there can only be a single gene. Okay? And then finally, just because there is an allele present doesn't necessarily mean that there is going to be an allele expressed. So this is the idea of complete dominance, right? If there is a dominant allele, that allele will always be expressed, even if there's a recessive allele present. This phenomenon of dominance and recessiveness is complete and absolute. There is no intermediate. Can you guys think of any characters within our own bodies in which that might not be true? That is, where A character isn't where there's a character that's a combination of perhaps mom and dad's traits. Yeah, what do you think, Carly?
Hazel eyes or brown and
That's a wonderful one. And I'm glad that you arrived there because my question was very poorly phrased. But eye color is a wonderful example of that. For eye color, you guys, there are two alleles. There's either brown or there is green. But we know that there are 4 eye colors, right? There is brown, there's green, there's hazel, and there's blue. And blue is an anomaly. We'll talk about that in a little bit. But what does brown eyes actually mean? What brown eyes means is you are homozygous for brown eye color. Green eyes means that you are homozygous for green eye color. And hazel eyes means that you are heterozygous. You have both a brown and a green allele, and they're both being expressed simultaneously. Okay. We'll talk more about some of those exceptions in a few minutes. Keep those 5 hypotheses in mind because we're going to find exceptions to each one of those as we move through this. So again, let's review what a Punnett square actually does for us. Punnett squares are incredibly useful tools because they model meiosis, they show us the types of gametes that can be produced, and then they allow us to calculate genotypic and phenotypic ratios. So if we were to look at this Punnett square, what we're saying is dad can, and let's use an anthropomorphic example. Let's say that dad can produce, and this isn't flower color any longer, let's say this is eye color, okay? If dad's heterozygous for eye color, what that means is he's hazel-eyed, right? So he has a brown allele, and he has a green allele. So what this Punnett square says is, one of dad's sperm cells will carry a brown allele, and one of dad's sperm cells will carry a green allele. And the same thing for mom, right? Mom will produce an egg cell that carries a brown allele, and mom will carry an egg cell, or produce an egg cell that carries a green allele. So this allows us to look at the kinds of gametes that parents are producing. That becomes infinitely more important when we start considering multiple genes simultaneously. So, you guys, what is the probability that dad is